नमस्ते बिटिया, हमेशा खुश रहो बिटिया। I am Dr. Bharat Sharma, Assistant Professor, Department of Pathology, Faculty of Homeopathic Science, JAYOTI VIDYAPEETH, Women's University, Jaipur. Today's topic is erythropoiesis. Hematopoiesis is production of formed element of the blood. Normally, it takes place in the bone marrow. Circulating blood normally contains three main type of mature blood cells: the red cells, also called erythrocytes; the white cells, also called leukocytes; and the platelets, also called thrombocytes. These blood cells perform their respective major physiologic functions. Erythrocyte largely concerned with oxygen transport. Leukocyte plays various roles in body defense against infection and tissue injury, while thrombocytes are primarily involved in maintaining integrity of blood vessels and in preventing blood loss. The lifespan of these cells in circulating blood is variable. Neutrophils have a shorter lifespan of 8 to 12 hours, followed by platelets with a lifespan of 8 to 10 days, while the RBC have the longest lifespan of 90 to 120 days. Their concentration is normally maintained within well-defined limits. Erythropoiesis is production of mature erythrocytes of the peripheral blood which take place in the bone marrow from morphological unrecognizable HSC. Red cell production is influenced by growth factor and hormones, notably erythropoietin. Erythropoietin. Erythropoietic activity in the body is regulated by erythropoietin, which is produced in response to anoxia. The principal site of erythropoietin production is the kidney, though there is evidence of its extra-renal production in certain unusual circumstances. Its levels are therefore lowered in chronic renal disease, while a case of renal cell carcinoma may be associated with its increased production and erythrocytosis. Significance. First is there is an increased production of erythropoietin in most type of anemias. However, in anemia of chronic disease, example is in infection and neoplastic conditions, there is no such enhancement of erythropoietin. In polycythemia rubra vera, there is erythrocytosis but depressed production of erythropoietin. This is because of an abnormality of HSC class, which is not under erythropoietin control. Erythro Erythroid series. First is proerythroblast. The earliest recognizable cell in the marrow is a proerythroblast or pronormoblast. It is a large cell, 15 to 20 μm in diameter, having deeply basophilic cytoplasm and a large central nucleus containing nucleoli. The deep blue color of the cytoplasm is due to high content of RNA, which is associated with active protein synthesis. Second is basophilic or early erythroblast. It is a round cell having a diameter of 12 to 16 μm with a large nucleus which is slightly more condensed than the proerythroblast and contain basophilic cytoplasm. Third is polychromatic erythroblast. Next maturation stage has a diameter of 12 to 14 μm. The nucleus at this stage is coarse and deeply basophilic. The cytoplasm is characteristically polychromatic, that is, contain admixture of basophilic RNA and acidophilic hemoglobin. The cell at this stage ceases to undergo proliferative activity. Fourth is orthochromatic erythroblast. The final stage in the maturation of nucleated red cell is the orthochromatic or late erythroblast. The cell at this stage is smaller, 8 to 12 μm in diameter, containing a small and pycnotic nucleus with darker nuclear chromatin. The cytoplasm is the characteristically acidophilic with diffuse basophilic hue due to the presence of large amount of hemoglobin. Reticulocytes. The reticulocytes are juvenile red cells devoid of nuclei but contain ribosomal RNA so that they are still able to synthesize hemoglobin. The reticulocytes in the peripheral blood are distinguished from mature red cell by slightly basophilic hue in the cytoplasm, similar to that of an orthochromatic erythroblast. Reticulocytes can be counted in the laboratory by vital staining with dyes such as new methylene blue or brilliant crystal blue. Reticulocytes are found normally in the peripheral blood. Normal range of reticulocyte count in health is 0.5 to 2.5% in adult and 2 to 6% in infants. Their percentage in the peripheral blood is a fairly accurate reflection of erythropoietic activity. The red cell. The normal human erythrocyte is a biconcave disc, 7.2 μm in diameter and has a thickness of 2.4 μm at the peripheral and 1 μm in the center. The biconcave shape renders the red cell quite flexible so that they can pass through capillaries whose minimum diameter is 3.5 μm through more than 90% of the weight of the erythrocyte consists of hemoglobin. The lifespan of the red blood cell is 120 plus minus 30 days. Red cell membrane. The red cell membrane is a trilaminar structure having a biomolecular lipid layer interposed between two layers of proteins. Important protein in red cell membrane are band 3 protein, named on the basis of the order in which it migrates during electrophoresis, glycophorin, and spectrin. Important lipids are glycolipids, phospholipids, and cholesterol. Carbohydrate from skeleton of erythrocytes having a lattice-like network which is attached to the internal surface of the membrane and is responsible for biconcave form of erythrocytes. Nutritional requirement for erythropoiesis. New red cells are being produced each day for which the marrow requires certain essential substances. First is metals. Iron is essential for red cell production because it forms part of the heme molecule in hemoglobin. Cobalt and manganese are certain other metal required for red cell production, while vitamin B12 and folate are essential for biosynthesis of nucleic acid. Vitamin C plays an indirect role by facilitating the iron turnover in the body. Vitamin B6, vitamin E, and riboflavin are the other essential vitamins required in the synthesis of red cells. Third is amino acid. Amino acid comprises the globin component of the hemoglobin. Hemoglobin. Hemoglobin consists of basic protein globin and the iron porphyrin complex heme. Normal adult hemoglobin constitutes 96 to 98% of the total hemoglobin content and consists of four polypeptide chains A alpha 2 beta 2. Small quantities of two other hemoglobins present in adult are hemoglobin F containing alpha 2 and globulin 2 globin chains, gamma 2 globin chains com- comprising 0.5 to 0.8% of total hemoglobin, and HbA2 having alpha 2 and delta 2 chains and constituting 1.5 to 3.5% of total hemoglobin. Synthesis of hemoglobin occurs largely in the mitochondria by a series of biomedical reactions. The reaction is stimulated by erythropoietin and inhibited by heme. Each molecule of heme combines with a globule chain synthesized by polyribosomes, a tetramer of four globin chains, each having its own heme group, constitutes the hemoglobin molecules. Red cell functions. The essential function of red cells is to carry oxygen from the lungs to the tissue and the transport of carbon dioxide to the lungs. In order to perform these functions, the red cell have the ability to generate energy as ATP by anaerobic glycolytic pathway, Embeden-Meyerhof pathway. First is oxygen carrying. The normal adult hemoglobin HbA is an extremely efficient oxygen carrier. The 4 units of tetramer of hemoglobin molecules take up oxygen in succession, which is turned result in stepwise rise in affinity of hemoglobin for oxygen. This is responsible for the sigmoid shape of the oxygen dissociation curve. First, in normal adult hemoglobin has lower affinity for oxygen than fetal hemoglobin and therefore releases greater amount of bound oxygen at partial oxygen pressure of tissue capillaries. A fall in the pH lowers affinity of oxyhemoglobin globin for oxygen, so called the Bohr effect, thereby causing enhanced release of oxygen from erythrocyte at the lower pH in tissue capillaries. A rise in red cell concentration of 2,3-BPG, an intermediate product of Am- Amden-Meyerhof pathway, as occurs in anemia and hypoxia, causes decreased affinity of HbA for oxygen. This in turn result in enhanced supply of oxygen to the tissue. CO2 transport. Another important function of the red cell is the CO2 transport. In the tissue capillaries, the partial carbon dioxide pressure is high so that CO2 enters the erythrocyte where much of it is converted into bicarbonate ions which diffuses back into the plasma. In the pulmonary capillaries, the process is reversed and bicarbonate ions are converted back into carbon dioxide. Red cell destruction. Red cells have a mean lifespan of 120 days, after which red cell metabolism gradually deteriorates as the enzymes are not replaced. The destroyed red cells are removed mainly by the macrophages of the reticuloendothelial system of the marrow and to some extent by the macrophages in the liver and spleen. The breakdown of red cell liberates iron from recirculation via plasma transferrin to marrow erythroblast and protoporphyrin, which is down to bilirubin. These are some common incidences. Red cell indices. First is mean corpuscular volume, PCV in liquid per liter, RBC count per liter. The volume value is 85 plus minus 8 mean corpuscular hemoglobin, mean corpuscular hemoglobin concentration. This session is powered by Digital Version 2.0, Jyoti Vidyapeeth Women's University. I hope you all enjoyed this session. If you have any query, please mention it in comment box. Thank you.